science fans, and welcome to SHE-ensya. In today's Molecular Biology and Genetics series, we will talk about how antibodies work. The world is now transitioning to the new normal, and in various locations all over the world, rapid test kits are being used to check if a person is eligible to go back to work or if they can now travel to different places. This rapid test kit is actually an antibody test that qualitatively detects the presence of IgM and IgG antibodies against the COVID-19 virus in human serum, plasma, and venous whole blood specimen for the auxiliary diagnosis of COVID-19 infections. But what is IgG and what is IgM? Both of these refer to unique molecules called antibodies that make up our immune system. There are actually 5 types of antibodies produced by humans. Immunoglobulin E or IgE are antibodies produced by the immune system if you have an allergy. These antibodies travel to cells that release chemicals causing an allergic reaction. IgD antibodies, on the other hand, are produced within immature B cells.
秘密兵器というと B 細胞のことですね。
その通り。早速現場に向かわせたまえ。俺に任せてください。こいつがあれば。
B 細胞、抗体産生細胞とも呼ばれている。
But despite the usefulness of B cells in fighting infections or counteracting allergies, the specific function of IgD is still generally unknown. IgA antibodies, on the other hand, are those secreted by our mucous membranes. This includes our lungs that produces, mucus known as sipon. The major protective role of the IgA antibodies is to bind microbes and macromolecules, to prevent or limit their binding and possible attachment to skin cells so that bacteria and viruses do not enter the body. And finally, we get to IgM and IgG. IgM is the largest antibody and it is the first antibody to appear in the response to initial exposure to an antigen from a virus or bacteria. IgG, on the other hand, is the most common type of antibody in your blood and other body fluids. These antibodies protect you against infection by remembering which germs you've been exposed to before. But how do these non-thinking molecules remember which germs you've been infected by before? Each tip of an antibody contains a paratope that is specific for one particular epitope on an antigen present in a virus or bacteria. The paratope in an antibody is like a lock that can be opened by a specific key, which is the epitope present in a virus or bacteria. And that is how antibodies can bind specifically, accurately, and with precision to only the virus or bacteria that they were created for. But considering the central dogma, how can an antibody made up of proteins adapt to new viruses or bacteria that the body may encounter? Aren't proteins coded for by pre-designated genes within the DNA? The lock and key mechanism of antibodies is made possible through programmed DNA rearrangements. These rearrangements affect the final structure of each antibody. And if you look at the general structure of most antibodies, it is often made up of a heavy chain and a light chain. The light chain contains a hypervariable region, which is the one that generally changes its shape to match a particular virus, bacteria, or molecule in general. Within the genes coding for these light chains are what we call leader and variable regions that are present in about 70 to 100 versions. Leader sections are indicated as L and variable sections are indicated as V. And together, they make LV. And yup, these code for the changing portions of your antibodies. Another portion of the light chain gene that has multiple versions is called the J region. The J region, which stands for the joining region, is about 6 kilobases away from the leader and variable regions. And there are about 5 possible versions of this. When the body needs to produce an antibody, one of the LV regions, along with its promoter, is randomly joined by a recombination event to one of the 5J regions. The recombination event eventually deletes the other versions that are not selected. It is this randomly selected version that is eventually translated into protein, and the version that is most efficient in attacking the harmful substance is the one that is eventually retained by the body. Our antibody diversity and the lock-in key mechanism are results of the random selection of a large variety of potential coding regions being joined together. Considering all that, why is it that it's the IgG and IgM antibodies that are being targeted for our rapid screening tests for COVID-19? This is because the body makes the IgG and IgM antibodies rapidly and immediately after being infected by a virus. So, these rapid test kits do not detect for the presence of the COVID-19 virus. They instead detect the antibodies that our bodies create when we come into contact with the COVID-19 virus. This means that the COVID-19 rapid test kits cannot be used as a standalone test to definitively diagnose COVID-19. This is because of the possibility that you could have the COVID-19 virus on your body but have yet to create the antibodies that are targeting it, or it is also possible that you have the antibodies fighting it due to a prior infection, but your body is no longer carrying the virus. The gold standard, or the more accurate test, is through the reverse transcription PCR kit. Reverse transcription PCR detects the presence of unique sequences of RNA extracted from substances in potential patients. These sequences are so unique that if they are not present, the primer molecules that target for their presence will have nowhere to bind, and the resulting procedure will come up with a negative result. Despite reverse transcription PCR being the best diagnostic tool, it still has a pretty high false negative rate at 1 out of 5 chances. This means that in 20% of the processes being done, false negatives can come up, which means that the data will tell you it's a negative, but the COVID-19 virus is actually present. But why is this the case? Molecular work is sensitive to fluctuations in temperature and inaccuracies in the volume of needed reagents. As little as a 0.1 microliter difference of volume could result to a failure in the polymerase chain reaction. The procedure requires a lot of training, and the more exhausted the scientists are when they perform these experiments, the higher the chance of errors in the results. Apart from this, there are errors that can arise from things we cannot control, such as fluctuations in electricity, different temperatures within the machine, and so much more. Automated kits where technicians need only to introduce the sample to the container with the correct reagents would significantly increase the accuracy of the procedure. But the costs for this is also significantly greater. We are experiencing the birthing pains of the new normal. But let us be hopeful that as long as the science and technology community get the support from the public and their local governments, we can eventually create cheaper, accurate, and more efficient kits that will allow us to diagnose viruses and diseases beyond COVID-19. I hope you were able to learn something from this short video on how antibodies are created in the body and how the rapid test kit of COVID-19 actually works. I hope you can give this video a thumbs up and also subscribe to our channel. If you have any comments, questions, or suggestions, please don't hesitate to message me, your resident Filipina scientist, in the comment section below. Thank you very much, and see you around!